Hello, everybody, and welcome back to another episode of the Lil Dudes Insect Academy podcast. I'm super stoked to be here in Washington, D.C. with my friend Tom Sappington. So we're just going to talk about bugs for a few minutes with you all. But welcome to the show, Tom. So go ahead and introduce yourself for everybody.
Yeah, thanks, Bradon. I appreciate being here.
Yeah.
So I work with USDA, the U.S. Department of Agriculture, the Agricultural Research Service, or ARS, in Ames, Iowa. And I'm a research entomologist. I study insect pests of corn, which is the mission of our unit there in Ames. But my specialty is insect movement. I'm an insect ecologist. So I am especially interested in dispersal and migratory behavior of adult insects. And they happen to be corn insects at the moment. I've done cotton pests in the past, butterflies.
Nice.
And actually monarchs recently, monarch
Very cool.
butterflies. So a lot of the questions that we have to answer related to how to manage pests, either to avoid resistance to BT corn or pesticides, or just understanding what they're doing so we can time control measures or whatever. They all depend on movement of the insects. And it's a kind of inherently a difficult thing to study because they're moving around, and we've got to find ways to figure out what they're doing. So we have different ways that we use and that I've used in my career. There's mark, release, recapture experiments where you mark the insects and then you know where you let them loose and you sample to see where they end up. Yeah. And you don't know where they were in between, but you know they went from point A to point B.
Right.
I've also used things called flight mills, where you glue a tether to the back of the insect and attach it to an arm that's on a post. So this is in a laboratory, and they can fly in circles. Right. So it's not exactly natural circumstances, but you can make comparisons between different groups of insects, like how do males compare to females and how far and fast they fly, or how do they change with age after a number of days after they've emerged from the pupa, things like that. So they're really invaluable for making comparisons, and you can learn things from flight mills that are really hard to learn any other way about what the insects are doing. We also use genetics, population genetics. So I've done that in the past, where you look at genetic markers in the insect, and you have a series of those. So you've got a profile of what they look like in this location, and then you compare them to what the profile looks like in another location, and you get a lot of these locations, and you can pick out which insects don't look like their neighbors in the surrounding, and then you ask, well, where might they have come from? And you can get a decent guess if you've got enough places sampled to tell where they came from. So there's times when you need to know that. Yeah. And it's a useful tool. And you can also get estimates of how far they can fly that way and so forth. So there's all these different methods. They've got different pluses and minuses for what you can learn, but when you combine them together, you can get a lot of times a pretty decent picture of what the insect is doing.
Interesting. Yeah. So what are some of the common corn pests that you've been studying recently?
So the ones we're working on or that I'm working on mostly are European corn borer, which is a moth, a small moth, but they lay their eggs on corn. They will lay their eggs in other places, but they really like corn the best. Right. And the larvae will bore into the stalk, and it disrupts nutrient flow in the plant, and the plant might even break over, makes it hard to harvest. So we study those. Also, western corn rootworm is a big problem. Yeah. In the Midwest or in the Corn Belt. Yeah. And that's a small beetle about the size of a ladybug. And the adults don't usually cause too much trouble. They can once in a while, but it's the larvae. They lay their eggs in the soil, and overwinter as that egg and the larvae hatch in the spring and feed on the roots of the corn. And the corn, that disrupts nutrient flow, and it also makes the plant weak, so a storm comes through and the soil gets wet and the plant lodges and tries to grow back, and you get this weird looking corn called goosenecked corn because they've fallen over and they're trying to grow back up straight. And that makes it hard to harvest, and the yield goes down and all that kind of stuff.
Yeah.
And they're just really hard to control because they've become resistant to a lot of methods over the years. They're just known for being able to dodge most control measures.
Yeah, and are these a big problem because they're invasive, or why are they a big issue?
So they used to be just another nondescript beetle out on the Great Plains, Colorado, western Kansas, and places like that. But around World War II, people started planting more corn, and they started growing it year after year.
Right.
And they can only survive on corn roots for all intents and purposes. So if there's rotation, then that's a good way to get rid of your rootworm problem, because they can't feed on soybeans or whatever you rotate to the next year.
Right.
When the eggs hatch, they're in a place they can't really survive in. But after World War II, they invented lots of insecticides that could be used to put down in the soil so that farmers could grow continuous corn. And a lot of them still like to do that for economic reasons. Sometimes they raise corn not to sell it, but to feed to their own livestock, and so that they don't have to buy it and that kind of thing.
Yeah.
So it is a big problem. It started spreading. I guess that's part of the point of this, is they started spreading out of that area on the Great Plains and all through the Corn Belt, and they're clear to the Atlantic Ocean. So they passed through Iowa, where I worked in the 1960s, I think, maybe early 70s, and they just kept going.
Yeah.
So they're invasive in that way. They're not introduced from a foreign country. They just expanded their range.
Migrated over time. Yeah. Makes sense.
Yeah.
Yeah, so let's go back to a little bit about you. Where did you go to school, and what was kind of that process over time, and what were some of the things you worked on?
So I went to undergrad school at a small university in Missouri, Central Missouri State University. And at the time, I grew up loving birds, and I wanted to be an ornithologist. Right. And found out there's not many places that hire ornithologists. Right. But I also learned as an undergrad that I really liked processes and ecology of whatever animal. Right. And I took a course in entomology and made a collection and thought, this is cool. I like this as much as birds, I think. And also worked for a professor there who he was an arachnologist, which studies spiders, and he had a big spider collection, like a serious one where he curated it and had it labeled and so forth. Wow. And so my job in that lab was to go in and he would come back from some expedition, or he had been on them in the past, and he'd come back with whatever spiders he could collect, just in a jumble, all in a vial, in alcohol. Yeah. And so my job was to look at them through a dissecting scope and separate them to what I thought were different species. I had no idea what they actually were, but that was a step that was a little bit time consuming that he wouldn't have to do, and I could do it. And, then he'd go back later and identify them, make the labels, and put them where they belonged in his big collection. Wow. So, and as part of that, and it sounds sort of trivial, but it wasn't, and it's, my job was before computers. I mean, there were mainframes, but nothing that no such thing as a personal computer. Right. So he had an index card system. He was trying to collect every paper that had ever been written on spiders or relatives, so arachnids, harvestmen, and so forth. But my job was to go through as they came in from, new research that had come in, and fill out an index card, the title, the author, cross-reference it with keywords, which meant I had to read the abstracts and try to figure out what this paper was about and what the important things were that were in that paper, so that he could look it up later. And so he had this master card file and find the paper. And he just had rows and rows of papers on bookshelves in his office. So that introduced me to what kind of research people would do with animals like that. You know, a lot of it was taxonomy, but a lot of it was ecological or behavioral, which was more what I was interested in. And so I started looking at entomology programs to go to graduate school, so I got a master's degree at Iowa State University. And there I worked for a USDA ARS professor. I'm actually kind of in his place now. He retired quite a few years ago.
Yeah.
But I'm in the same unit and all of that. And working on European corn borer. So, and it's an agricultural pest, and it was just kind of a random chance that that's the position I took. I had a couple other offers, but that looked most interesting. I knew nothing about agriculture whatsoever.
Right.
Didn't grow up on a farm. I could tell what a corn plant was, and that was about it. So I learned a lot about what farmers care about and so forth, and why my research mattered in the long run to them.
Right.
You know, other scientists would use that information who were also trying to help the grower and so forth. So that was a good experience. Then I went off and worked for 3 years with a crop consulting service where you go out and you scout fields and look for insect problems.
Right.
But as well, also weeds and fungus problems, and you write reports and you explain to them what they've got. And it was just ironic that I knew nothing about agriculture trying to advise farmers, and I had to get a lot of help with the questions they asked.
Took some learning.
It did. It was, and I wasn't cut out for that. I learned that. I mean, I did well, and the owner of the company tried whatever he could to keep me on, but it's like I wasn't that interested in that angle because it wasn't so much biology as it was agronomy.
Right.
So I went back to graduate school at University of Kansas, and there I worked with Chip Taylor, who is still around, and he's the one who does Monarch Watch, if you've ever heard of that. He invented that, so that's where people tag monarchs and they look at their migration. Yeah. So he hadn't organized that until right about the time I left, but I was working on a different butterfly, which was coleus butterflies, which are orange sulfurs. They're real common in alfalfa fields. Okay. You can see them around just about anywhere in the U.S. Right. And I was doing behavior experiments on them, going out in the field and capturing them and looking at the pheromone that the males had on their wings. So they would court females, and the females are evaluating the males that want to mate, and they're looking at the colors on their wings. They can see ultraviolet, which we can't, and they have ultraviolet patterns on their wings. So the females test that, but they're also testing this pheromone that's on the scales of the hind wing of these males, and they're beating around the females' antennae, and they can sense that. But they're variable, and there were like 3 components. The person who was there before me had figured this out, the grad student before me. So I knew what I was looking for. I had to learn gas chromatography, which is a chemical process where you just shoot stuff into a column. It goes through, I say a column, but it's coiled, so it's like 50 feet or something of this column, where it separates the chemicals, and you get these peaks that come in the readout. Okay. And you put in known chemicals, so you can use that to judge the position of what you're looking for. And then you can measure how much is on the wings. And I was looking at ratios of these different components and trying to figure out if the females were using this, because we knew from observation that females would accept some males and reject others, but we didn't know why. You know, what are they using to choose? Right. And found out that they are using some variation in this pheromone. And sulfur butterflies are interesting because the females come in 2 forms. They can either be white, or alba, it's a Latin word for white, or orange. And orange is more common, but about 1/4 of them are white. And they were choosing different patterns of that courtship pheromone. So the white ones liked a different pattern than the orange ones. Still don't know why that is, but
Interesting.
but that was a really good project.
Yeah.
And I went back to Bill Showers at Iowa State, who I got my master's from, and did a postdoc there and studied migration of black cutworms, and that's when I figured out how to tether them and put them on flight mills, and was looking at their migratory behavior.
Yeah.
And there are species that they're kind, you think of birds migrating. They, go south in the winter and north in the spring, and there's some insects that do this, and black cutworm is one. They can't overwinter in the north, and yet there's all this great corn up there and other plants. They actually like different kinds of weeds better than corn.
Yeah.
But they get in corn and cause problems. And they go south in the winter and overwinter there. They can't go into diapause, which is like hibernation.
Right.
And then they migrate back in the spring. And it's not the same individuals because they don't live that long, so it's considered multi-generation migration.
Right.
And they know to go north in the spring. They probably use the Earth's magnetic field as part of how they know which way's north. They get in very fast winds up high, so a lot of migratory insects do this. They'll ascend into the atmosphere. Moths will do it usually at dusk. That's what cutworms do. And they go up, they can go up to 2 kilometers high, but they're looking for fast winds moving in the right direction.
Right.
And so in the spring, they're called low-level jets that go in front of the cold front, and they'll move north really fast. And in the fall, it's a different generation that they come back. It's a lot slower because the winds aren't as fast. It may take them several days.
Right. So it's a step-by-step, generation-by-generation migration.
Yes.
Interesting.
Yeah. So I did that, and I decided I was still having trouble finding a permanent job. So I took a postdoc. I figured, and this was at the time where molecular biology, including in entomology, was really taking off.
Yeah.
And a lot of labs were doing that. And I thought I need to get those skills.
Right.
So I took a job studying mosquitoes at Michigan State with Alex Reichel, was my mentor, and was looking at physiology of egg development. And that's a long story, and it was a really steep learning curve, and it was, I was wondering if I'd made the right decision, because it was like almost starting from scratch. I was learning things I had very little background on, and it's a complicated field. You've got to learn the techniques and so forth. But it turned out okay, and it was really good. And then from there, I got a permanent job with USDA ARS down in Weslaco, Texas, which is right on the Rio Grande River. It's very deep south, kind of between Brownsville and McAllen, if anybody knows where those cities are. And the mission of the unit there was cotton, so I was studying cotton pests and movement. It was mostly short-range movement, but we also started using population genetics. So that's when the molecular stuff I learned from Michigan started coming in handy, because I could at least understand what you could do to do that kind of thing. So, and the thing about boll weevil is they had this big eradication program, and they started in Virginia or somewhere, and were pushing east, and the eradication was mostly just nuking the heck out of every cotton field and with insecticides, driving the populations down to zero. And we got lucky; the boll weevils never got resistant to the insecticide they were using. And they were pushing through Texas when I was there, and so, but these eradication zones were of several counties, and they had to be voted in by the growers. They had to have a referendum because they were the ones who were going to pay most of the costs. And not everybody was on board with that. And once it became law, everyone had to participate, and everybody paid a fee. And so the thing is, you get these zones, and some of them enter at some times different than other zones, and you get eradication in different different stages. So you
Right.
might have a border of one zone where there's still a lot of boll weevils, right next to one where they've almost been eliminated. And so they
Right.
needed to know how much movement they could expect between boll weevils, because
Right.
reinvasions are really expensive to get rid of.
Right.
So that's what I was working on, and we used population genetics and showed that they were moving a lot farther than anybody suspected.
Yeah.
And that, and things like they'd get a reintroduction, say, in the Kingsville area, and then they would blame it on trucks that were coming through, or contaminated equipment.
The
But we
trucks
could-
were transporting them. Yeah.
Or on somebody's passenger vehicle, or
Anything,
just
yeah.
anything. But what we were able to show was that, they're using population genetics, is that they're flying up. We can tell which ones are immigrants and where they're coming from.
Right. Yeah.
And we could backtrack it, not me, but colleagues could backtrack weather systems and show which weather systems they used to show up at the time they were getting them in the traps
Wow.
and all that sort of thing.
Yeah.
And so that made a difference, because there was no reason anymore to put quarantine measures on the Rio Grande Valley, to forcing vehicles to be power sprayed and stuff to prevent
Right.
weevils from going up, because that's
Right.
not how they were getting there.
Yeah.
They were flying on their own.
Yeah.
So I was there for about 4.5 years, and then a professor who was on my committee, who, as a master's student, remembered me, and he needed an entomologist, and just called me up and asked if I'd want to come back. Our family's up here and all that, so came back to Iowa, and that's where I've been since 2003,
Very cool.
and studying the corn insects.
Nice. Yeah. So I think you covered pretty much all kinds of stuff. But apart from entomology, or maybe related to entomology, what are some other hobbies that you enjoy? Some other activities that you like in your spare time?
Sure. Yeah, and I still like to bird watch.
Mm-hmm.
Keep a list. I like to play chess. I read a lot, especially history,
Nice.
and I like to write, stuff besides technical papers, which
Right.
I do for a living, but,
Yeah.
but I like to write other stuff as well.
Yeah.
So things like that. I like to travel. I've been to a lot of different places. As an entomologist, that's one of the perks, is you get to go to conferences like this.
Get to go to cool
that
places. Yeah.
you might not get to go otherwise.
Right. Very cool.
Yeah.
Yeah. Okay. So, what are some of your goals and aspirations for the future? Like, any cool trips, or any research that you want to do moving forward? What are your goals for the future?
So my future is I'm going to retire in about 9 months, and I
Nice.
thought of this last night. 9 months is like the gestation period, so I'm going to be born into a new world in about
Nice.
9 months. And so I won't be a professional entomologist at that time. I'll be retired professional entomologist, and I'll still kind of pay attention to what other people's
going on.
research is going on, but I won't be doing any actual research of my
Right.
own anymore.
Yeah.
And we'll spend time, I've got grandkids, and we'll be traveling, and I'll be trying to write,
Nice.
non-creative nonfiction, which is actually a thing.
Very fun.
Yeah, and I do have one experiment left to do with flight mills before I hang it up, and that I'd like to get that paper written up and
Yeah.
put in whatever I find
Yeah.
out with that.
All right. Well, where can we learn more about some of the work you've done? Is there anything online, like maybe social media profile that we could go to, or a website, maybe just for some of the labs you've worked for, or for the USDA? Is there anywhere that the viewers can go to learn more about some of the work you've done?
So I don't do social media because I'm kind of a dinosaur that way. But,
I totally understand that.
but I did do a paper with a grad student just a few years ago that's it's actually a visual video paper.
Yeah.
And I can give you the link to that, and it shows him how to tether rootworms. I'm on there a little bit talking about some background.
Yeah.
So that's a good thing to see what we do in action. There's some other links when they had the International Congress in Florida in 2016. I was on one of the organizing committees and stuff, and I had been to several of those congresses in the past. They only meet every 4 years.
Mm-hmm.
And when I was going to those, I would write letters home to my wife because she couldn't go. She was home with the kids. But I wrote them in a way to make her feel like she was there.
Yeah.
And so I published some extracts of those on Entomology Today, which is ESA. It was new at the time, but ESA's blog site.
Nice.
So I've got some links for 4 of those. One was, so it was Italy, Brazil, Australia, and South Africa were where the meetings were. And
Very
on
cool.
those trips, I would usually take a week either side of the meeting to just go see things.
Yeah.
Because I was in an exotic place. And,
Cool. Yeah.
so that would give a feel for. And the extracts that I took out had to do mostly with entomology type themes.
Right. Yeah.
Going to talks and things. It's still pretty conversational. And anyway, so I'll give you those links.
Yeah. Okay. So this was super fun, Tom. It was so nice to meet you. Yeah, this was a great interview. So thank you so much for being on the show again. This was really great.
Yeah. Thanks, Bradon. I really admire what you're doing.
Thank you.
Podcast.
Yeah. Thank you so much for listening to this episode of the Lil Dudes Insect Academy podcast. If you enjoyed it, definitely consider dropping a follow on whatever platform you're listening on. And also, Lil Dudes Insect Academy is a non-profit organization. So if you're passionate about science communication, entomology, outreach, and education, and you enjoy what we're doing and how we're solving that gap, then consider heading over to our website and donating there, or becoming a monthly donor via our Patreon, which is also linked in the show notes of this episode. Yeah, so share this episode with one of your friends, and tune in next week. I will see you all next week, and with that, keep on bugging.